Hello class. Today I will be talking about meiosis. Woo! Meiosis. So meiosis is just pretty much DNA replication for new cells, but they lean more on the side of sperm and egg cells, which is we call them gametes. So we don't have to always specify sperm and egg cells. There's a lot to say. So the first stage is called prophase. Prophase has to do with the nuclear envelopes. When the chromosomes from the mom and dad pair together like this, they pair together to create a homologous pair. And notice how each chromosome, Notice how each chromosome has a different color on each side. That represents that the chromosome they're sharing genetic information, and that genetic information is from the mom and the dad, and that's called crossing, or crossing over, I should say. Okay, so the next stage after prophase is prometaphase. So prometaphase is when the spindle fibers, which is the white Play-Doh that's right here. The fiber has these string-like fingers that are called, it's a pink fiber. They're called microtubules. So the microtubules are connected to the chromosomes. And what's connecting them is this protein called a kinetochore. And all that happens in prometaphase is that they're attaching to each other. That's all that happens in prometaphase. And as you see in the next phase, they're going to start moving and shifting and doing this, that, and the third. All right, so this part is called metaphase. All right, so during metaphase, the homologous chromosomes are arranged in a pair. So that means they come together like this, and they pair up together. And when they pair up together, they pair in the middle, straight down the line. This is called a metaphase plate, which is just pretty much saying it's the middle of the cell. That's all it is. However, what helps the chromosomes get to the middle is microtubules. They help align them. Yeah, they help align them into the middle of the cell. All right, so the next step after metaphase is anaphase. And anaphase is just, during anaphase 1, the microtubules, which are these right here, the pink fluorescent looking things, are pulled apart. Yes, pulled apart at opposite sides of the cell. And I will explain why in telophase. Okay, so for this step, it's called telophase with an additional cytokinesis. So in telophase, during telophase 1, the chromosomes are pulled to opposite sides like they were before. They're pulled to opposite sides. Now, they're pulled to opposite sides of the nuclear envelope, and they are decondensed. And because they are decondensed, the envelope is loose. And when the envelope is loose, it starts to form another nuclear envelope. And with that forming of the other nuclear envelope, they actually separate and divide. And then there are 2 cells. Look at that. Woo! 2 cells. And that process of splitting is called cytokinesis. But hold on, because it looks a little bit more disorganized than that when they split. So when they split, this is how it usually looks. Because they're decondensed, they're falling apart. All order is breaking loose. So this is how it would actually look, because it's broken apart and they're all squiddled all up again. Now, after this phase, we're going into meiosis 2, which is just another form of separating. So now we are in meiosis 2. Meiosis 2 is going to, it's a second part of the replication process for gametes and such, and sperm cells, egg cells. This is the second process of that. So prophase 2, all it is that the chromosomes are condensing again after being, these chromosomes are going to condense again inside this cell because they were out of order. Once they split apart, they were out of order. Now they got to condense together again. And when they condense together, they're going to make a better structure. They're going to make a better structure that's better for prometaphase 2. Well, with the help of prometaphase 2. So we are in prometaphase. We are in prometaphase 2 now. And prometaphase 2 is the spindles have formed on both sides, which I'm going to show you now. And we're going to discard this cell because we're not using that. But this is what will be happening in prometaphase 2. So these cells, so the chromosomes will be lining up again in prometaphase. They will be lining up again, and they will be attaching to the spindles. Because the spindles have formed, everything's going right. The spindles have formed, and the microtubules are attaching to the chromosomes, and now they are aligned. They are aligned in, it doesn't want to line up. They are aligned in the middle. Well, they are aligned in the middle. And the spindles have fully formed, like I said. Each sister chromatid they're attached to the chromosome. But as you will see in metaphase 2, what's going to happen to the chromosomes. So now in metaphase 2, the microtubules are connected to the kinetochore, which is it's like a little pink thing here. That's the protein that the fibers, that the microtubules, which are these things, connect to. And now they officially align in the middle, kind of like what was going on in anaphase, but actually not anaphase, what was going on in prometaphase, but prometaphase is more just they are attaching, I should say, instead of just linking together. They're just attached in prometaphase. But in metaphase, that's when they actually are connected and aligning together, as you can see. So this is anaphase 2 now. Well, we're in the stage of anaphase 2 now. And that is when the sister chromatids are being pulled apart by when the chromosomes are being pulled apart into sister chromatids by microtubules that are being connected by the kinetochore, which is that pink thing right there, which would be the pink thing. And then they move to the opposite side. So I'm gonna show you the tearing of that and what it would look like. So they would tear, and they would be left with just the one chromatid. They would tear from each other and be left. They would be left with one chromatid, as you can see here. Left with one chromatid. So now we're in the last stage, which is telophase 2 of meiosis 2. So during telophase, the chromosomes are at completely two different ends of the cell. Well, yeah, they're at the two ends of the cells. and the cell will split once again to make a new cell. And when they split, just like in telophase 1, they're going to split, and the fibers and everything that was the second part of this, everything that was in here is going to go to the second cell, just like how it did in the first telophase. And now they are split, and now you end up with a completely two different but matched up well cytoplasms. And now we end up with non-identical cells at the end of telophase 2.